But I will be searching those and showing them to you, sharing my screen. Okay, but then we will look at an onion cell or onion cells underneath a microscope right here. I have a microscope cam, and I'm hoping that it works. Okay, good with that? Zach, we just went over it. I will have to meet with you and talk with you about where to find it. Okay. Those of you that are not sharing your screen, that is of course okay, but do be aware that I have you on relay, meaning I can see your screen. So if you're not paying attention, if you're on YouTube watching stuff on mute, I can see that. All right. Not saying that that's happening. Just be aware that that could happen. All right. We cool with that? Cool. All right. So you should have watched a video the other day from Mr. Welch teaching you about the microscopes, okay? We are going to review that. So some of you, this will be your second or third time seeing it. That's okay. It happens. And I gotta know where to find it. Oh no, I know where it is. Hello. The microscope discussion. Okay. The microscope discussion, you should have completed that. Most of you did, I think. I guess I haven't seen anybody who's had any trouble. So if you are looking for that now, you would need to go into older stuff. So just a quick breakdown, and you need to give me feedback if Schoology is too confusing. I will try. I'm trying to make this not as confusing. But up here would be a recent week, the week that we're in. This is on-demand lessons where I put recorded lessons. There are some in here, okay, from our labs and whatnot. Then you would go into, if you are missing things from back in the day where you're like, oh, I need to turn this in, I have a 0, it would be in older stuff. Okay, older stuff, because that's the best name I could think of at the time. We have agendas from earlier weeks. We have discussions from earlier weeks if you need to catch up on those, which some of you do still. All right, so then I would go to microscope discussion. Here is where we're going to listen to Mr. Welch explaining the parts and functions of a microscope. Okay? Okay. And now I know it went to a new tab, so I will have to share a different part. Okay? This is the part where it gets just tedious. Entire screen. Okay? So we're going to go over it. I'm going to talk as well. Cool? Everybody see Mr. Welch on screen? Yes? Okay. Mr. Welch taught science here at the middle school for many, many years. I believe he was at the high school as well, some points. He's fantastic. Unfortunately, due to COVID this year, he may not be subbing as much or at all. So, but if he does, listen to this guy. He knows his stuff, and he's really cool.
Your microscope should look like this. It should be stored away with the cover over all glass and above the base and below the stage. When you take the cover off, and you fold it neatly, and you set your cover aside. The microscope, if it was put away properly, should be in low power objective. The stage should be all the way down. The eyepiece that is movable should be over the arm of the microscope.
So he just named about 4 parts right there. The eyepiece, the arm, the base is what it just rests on. The stage is what you put your slide on. The arm is what you grab it by, and the base. And then you have, of course, the eyepiece, which you look through. Okay? So he named about 4 pieces. If you didn't do the discussion, this would be very helpful for you.
The cord should be wrapped tightly, so it's wrapped up on the back side like this, so it's easy to carry. When taking it out of the cabinet, it should be covered, and you should be grabbing it by the arm and supporting it by the base and carrying it close to your body and your elbow should be in when you come to your station. You put your microscope approximately 1/2 width of your hand away from the edge of the table when placing the microscope on the table before beginning to work. You put your case aside. Now it's time to plug your microscope in. So you unwrap your cord gently. You never fling it around. You just then plug it into the plug. We have turned the light on to be sure it's working. Now it's time to go over the parts. We have the eyepiece on the movable head of the microscope. This allows the teacher to be looking at it from this end and for you to be looking at it this end. If you'd rather load your microscope differently, you can turn your microscope all the way around, turn the eyepiece to be working this way, and slide your microscope slide in this position instead. But most people work with the arm in the way and just work this way over the top. Okay, so this is the eyepiece, this is the head, this is the arm, this is the movable stage, this is the revolving nose piece that supports the 3 objective lenses. So this is the eyepiece lens, and these are the 3 objective lenses. The smallest lens is the shortest, and this is the less magnified lens, and that is low power, and that is 4 power. 4 times 10 would be 40 magnification. So the eyepiece is 10. This objective lens is 4, so 4 times 10 is 40. If we move it to medium power, that would be 10 power here and 10 power here. That would make it 100 magnification. You multiply the numbers. When the teacher says you may move it to high power, you get your nose down to the stage and you move it into high power. It clicks into place. You'll know light is passing through by looking through, and you have a complete circle of light coming through your eyepiece. And now this is 40, so 40 times 10 would be a magnification of 400.
So, Mr. Welsh, why would somebody want to have their nose down before they put it into high power?
Good question. You want to have your nose down to the stage because when you have your glass slide in place, the stage would be up here when it's under focus, under low power, and you're up like this. And when you move into medium power, you can see the distance between the medium power and the slide that would be in place would be a lot less. And now when we move to high power, it's just barely above the glass. So we don't want the lens broken or the slide to be broken. So we have to be sure there's nothing that's causing problems where the lens is touching the cover slip or the glass. So if there is a problem, you have to back up and go back to your original low power and ask for help from your teacher, and then we'll make some adjustments, okay? On top of the stage, we have stage clips that clip down our glass slide. We have 2 adjustments. We have a coarse adjustment up here on top, which allows me to move the stage in great amounts of movement. And then below that, we have a little fine adjustment. So if I bring this all the way up and I now need to get a better focus, I grab the fine adjustment, and you can see it's barely moving the stage. But this one moves the stage in great amounts, and this moves it in a lot less amounts, okay? We have then time to clean up. If it's time to clean up and I have a slide in place, I always have to bring my objective lenses back into low power first, so there's plenty of space between the slide and the lens. Then I take my coarse adjustment and I move it down, and then I can take my slide off. Now, if your teacher says we are working in high power now, so I'm going to move it to high power. And now it's time to clean up. We cannot touch coarse adjustment because it's in high power. If we did, we could ram the stage up into the lens and cause major problems. So whenever we're in high power, we're always working with the fine adjustment, the smaller knob. If we want to work with the coarse adjustment, we have to move back to a medium or to a low power. So medium and low power, coarse adjustment. Medium power, coarse adjustment. High power, only fine adjustment. Now it's time to clean up. So I'm moving back to low power. I'm lowering my stage all the way down. I'm turning my light off. I'm unplugging my cord by the plug, not by the cord. I'm rewrapping my cord in position the way it should be, ready to be stored. And we should then be in low power, stage all the way down, clips in place, eyepiece over the arm, light off. We take the cover and we put it over the top of the glass materials first, but not under the base. It should be under the stage like this when stored. And then again, we carry it back to the cabinet by grabbing the arm, supporting the base close to the body with our elbows in and carrying it back to our position. And putting it under the number, we have number 6 here, so that would be our number 9. That would go in slot number 9 on the cabinet. Any questions?
Oh, that sounds great, Mr. Welsh. Thank you for explaining how to use a microscope.
All right, so there you have it. That's Mr. Welch. If you ever come back in person, you are good to go. There we go. So that is that. Okay, so here's what you need to accomplish today. We are doing a cheek cell versus onion cell lab report. And you can find that on Schoology. Ready? I'll show you where. Present now, a Chrome tab. Let's go. Alrighty, so here's where you would find it. We are in week 5. We are looking at Thursday. Here is your cheek cell versus onion cell lab report. You would click on this. Okay. Then.
We can't see your screen.
Interesting. Let me fix that. Thank you for letting me know. Mr. Dayton, figure it out. Schoology. There we go. So this is for on-demand learners too, who are watching this. Okay. There we go. Here's where you would go to do this lab report that we're about to do. Okay, go to Thursday, October 1, 2020. Cheek cell versus onion cell lab report. That's where I'm at. And then I click on this, and this template is already made for you. Okay, I need everybody to get there within the next 30-ish seconds. When you are ready to go, put a pencil behind your ear. If you have one, you should have one, because you're going to need to be drawing too. When you are at this, when you're at that, whatchamacallit? The lab report, you're good to go. So again, I would click on this right here in the Thursday, show everybody again. I'll show everybody again. We are going to science. We're going to week 5. We are going to Thursday. We are going to cheek cell versus onion cell lab report. Then once you're here, you would click this, and it'll take you there. Yep, I will repeat them. Here we go, people. All right, so here we are. You are in Schoology, you should be. You are in your science course, or you should be. All right, then you will go to week 5. That's the week we are in right now. And it's now not loading. Okay, there we go. So I'm in week 5, September 28th through October 2nd. Then I click on Thursday, October 1st. Oh my goodness. Are you able to get there? Because mine doesn't allow me. Is
Yeah,
Schoology
mine just
work?
said that.
Is Schoology
I can
working?
get there.
Yeah,
Yeah,
it
I
does
can get
work.
there.
This is gonna drive me insane if Schoology does not work when we need it to work. So I'll click on Thursday. Oh my gosh. All right. This is fine. We'll figure it out. It sounds like some of you were able to get there. I'm going to do it again to see if I can get
There is a link, though. I couldn't find a link underneath it.
Was that you, Cooper, who said that? Who said that? Zach?
Yeah.
A link to what, buddy?
So, like you said, you showed us in the first time. Whenever.
Are you able to get to this lab report? That's it. That's all I need you to get to.
I guess I can get there. Yeah.
Okay, good. That's what.
I can get there.
Okay, good. Well, then I'm not going to monkey with it anymore. If you cannot get to that lab report, I refreshed it, James. Thank you for that advice. I'm just going to not monkey with it anymore. There we go. So that's where you need to be. All right. That's where you're typing in your name, your period. I would just say your live remote, and then your date is 10/1/2020. Everybody take this time to type that in now. Cool? Yes, Molly. Question?
My Schoology won't let me in. It says it's temporarily unavailable.
Yeah, that's what I'm kind of running into right now, and that is incredibly frustrating if that is still the case. Yeah, it's not loading very well for me right now. So what I would do if I were you is to just kind of follow along with us right now. Okay, and maybe jot some of these things down that I'm talking about, because you can see my screen, right? And I'll take you through what you need to do. So if you want to write some of these things down, you can. Okay, we're gonna move
Mine
on.
is.
Megan says the same thing. Okay, Megan, follow along with us and we should be good. Just follow along and write these things down. Okay, we're moving on. If you have problems, just let me know and we will have to figure it out later. So here we are. Your name should go here. Your period should go here. Your date should go here. Everybody should type that in within the next 30 seconds. You should be able to do that. So I'll give you this time to make sure your name, period, and date is on there. While I'm doing that, be thinking about a problem and what it needs to be in the form of. Yes, James, I will tell you the Benedict solution materials. I believe when I edited your lab report, I believe I put them in there. I'm not going to check right now because we're doing this, but when I get a chance, I will check that. I believe if you check your submission, unless I didn't submit it back to you, it's on there. Okay. Cool. All right, 30 seconds is up. Now, you need to be thinking, what is the problem always in a form of? Alton, what do you got?
A question.
Yeah, it's a question, so it must end in a question mark. If you look at this lab report, you will see that the P space is blank. That would lead me to believe that you need to come up with that problem, right? That's what you should be thinking. Now, you're probably thinking, well, how am I going to come up with that? Well, let's take a look at the hypothesis that was already given for us. Okay, Mr. Mallon and I already came up with a hypothesis for you. You didn't get a choice in this case. Sorry. Okay, so for our hypothesis, I'm going to read that to you. Whistling needs to stop. Whistling needs to stop. Not sure who it is. Can't see, but whistling needs to stop. Thank you. All right. So hypothesis, if I look at both an onion and cheek cell, then I will be able to see that the animal cell or cheek cell is larger, but both have the three main parts associated with all cells. Okay? So if I look at both an onion and cheek cell, then I will be able to see that the animal cell or cheek cell is larger, but both have the three main parts associated with all cells. Okay, so I've already told you today that we're looking at both an onion cell and a cheek cell. A cheek cell is an animal cell because you are an animal. All right? We are made up of animal cells. Onion. What is an onion made up of? Go ahead, Sawyer.
It's made up of food.
Yep, good. I would eat an onion, but what do you have to do to onions? Where do they walk around? Do they hunt their own food? Cooper, what do you think?
They just kind of grow on a plant. Not
Yeah.
sure what. They grow off a plant.
Yeah, sorry for interrupting you. They grow on a plant because they are a plant, right? So they are made up of plant cells. Plant cells and animal cells are different, okay? So we're going to see how they're different today. You need to come up with a problem that can be answered this way. So if I look at both an onion and cheek cell, I will be able to see that the animal cell is larger, but both have the three. That was the hypothesis that we gave you. What question does that answer? Or what question does that attempt to answer? Anybody? Alton, let's hear from you again, man.
Which cell is larger?
Sure, that's a good one. Which cell is larger? Or what I would even accept, what are the differences between plant cells and animal cells, right? Or how can I find the differences between plant cells and animal cells? Those were three good questions for problems that you could write right now. You could type right now. Those of you that don't have access to this document, you should be writing that down for your problem. But otherwise, you should be typing this, people. All right? So here's questions that are problems that you could answer in this experiment. How are animal cells and plant cells different? How can I tell, or how can I find out the differences between animal and plant cells? Between animal and plant cells, which one is larger? Okay, those are three examples. I'm giving you 1 minute to come up with a question. I gave you three good examples. Okay. Alrighty, that was that. That was a minute about. Yes. If you have a microscope, sure, Alton, you could give it a shot. Do you have an onion? You do have a cheek. Yeah. Do you have toothpicks? Okay. So if we were to, then we're going to get into this, and I'll tell you how you could do the experiment if you wanted. Because it really is not that difficult if you have a microscope. Okay. So we're moving on. The hypothesis is there. Our materials, here they are. We have a microscope. I have a slide. This is a slide for those of you at home. On top of this slide, you might not be able to see it, is a cover slip. The cover slip is the thing that I put on top of my specimen that I'm looking at. It's plastic. The slide is glass. Okay. The slide is glass. The cover slip is plastic, and it covers whatever I am trying to look at. so that the microscope doesn't touch it. Then I have onion as one of my, that is already on here. I have that put on here. Toothpick would be normally used in this. I'm not this year because of COVID. Cheek cells, again, not using because of COVID. Water, we don't need right now either because we're not dealing with any of that. Iodine, we are also not needing. So all of these right here, I could strike through those. Format, text, strike through. Okay, those we are not using this year. Okay, we are also not using methylene blue, which I am about to strike through. Format, text, strike through. Okay. We will be using, oh, sorry, I did not mean to cross out iodine. Figure it out. Okay, we are using iodine, excuse me. So you'll notice that this is stained. That is iodine. There's an onion, piece of onion skin, piece of onion under this, and that is stained with iodine. Why do you think I stained it with iodine? Laylee, why do you think I stained the onion skin with iodine? I could not hear you. I don't know if you said anything. Yeah, I still can't hear you. Sorry. We'll move on. Thank you for attempting to answer. Let's see. Julia, why do you think I stained the onion with iodine? Julia looks frozen. Okay, maybe third time's a charm. How about Sawyer? Why do you think I stained it with iodine?
So you can look at it easier.
Yeah, pretty much. So I can see it a little bit easier. Good. Onions are white. The skin, if you cut it really thin, is kind of clear. Harder to see under a microscope. The iodine is an indicator to help us see. Thank you for that. Third time was the charm. Okay, so moving on to methods. We will talk about that after we do this. Now we're moving on to observation. Okay, observation. First, you are looking at an animal cell. Now, I'm going to quickly scroll all the way to this rubric. Okay, here's what it takes for you to get a 3 in 2 targets on this lab. This lab is 2 targets. All right. So science target 1, in order to get a 3, your question is clearly defined and related to the hypothesis. So if your question is related to your hypothesis, you're in good shape. You'll get a 3. Correct punctuation is observed, meaning it ends in a what? Your question ends in a what? Cooper made the question mark symbol. Yes, thank you. It is a question mark. I appreciate that. Okay, then in order to define your problem, your conclusion must hit all 3 parts and clearly answer your question. Remember, your 3 parts of a conclusion are summary of results, whether or not your hypothesis was correct, and self-critique. Okay, so we're moving on. I will write that down right here, what you should include. Summary of results, whether or not your hypothesis and self-critique. Okay, all right. So moving on to observations, or moving on to what it's going to take for a 3 in target 2. Okay, images are clear and take up the entire space. So you're going to need to draw. Obviously, you can't draw on this document, but you're going to need to draw and take up a portion of paper that would be this entire space, drawing what you see. Okay? Then, another part, labeling is consistent through all three powers. We're going to do 2 powers because I'm not going into high power today. So, what do I need to label on a cell? What are the 3 main parts of a cell? James, we'll get to it. I have a microscope here. We'll get to it. What are the 3 main parts of a cell? We talked about this. We had a lesson. I know we did. 3 main parts of a cell. Molly.
The 3 main parts of a cell are a cell membrane, a cytoplasm, and a nucleus.
Yep, cell membrane is the membrane around the cell. Cytoplasm is the thing that the jelly-like substance that fills the whole cell, and the nucleus is the control center of the cell. You will need to label those 3 things in your drawing. Okay? Then, this is an important one. There is a noticeable difference in between the images and powers. So if you draw an animal cell on low power, and then you draw an animal cell on medium power, I should be able to tell the difference in those powers. It should probably, actually not probably, it should definitely be larger in medium power. Does that make sense? That shows me that you are understanding the difference between low and medium power. You will also need to label the nucleus, the cell membrane, and the cytoplasm. Okay, then finally, methods are completed with clear instructions on what was accomplished. We will maybe get to that. Okay, so let's move on to observation, okay? You should have a pencil, you should have a piece of notebook paper that you are drawing in. You are drawing an animal cell under low power. Ready? Here we go. So, I will put this, can you all see this?
Yes.
Okay, thank you. I'll go back to it. This is a cheek cell under low power. It's kind of blurry. Why don't I go to this one? Here we go. So these bluish things, we would normally look at cheek cells stained with methylene blue to help see them because your cells would be very tiny and clear or transparent, and they're hard to see. But here you can see there's like 1, 2, 3, 4, 5, 6, 7 cheek cells, along with some gunk from this person's mouth. All right, perhaps bacteria. This is what you should be drawing right now on your notebook paper that's about the size of that square on your lab report. Okay? Then you should label these. All right, let's take a look. I would label 1 cell, but I would be drawing these. Okay, so if I look at this cell right here where my hand is, all right, I'm going around this cell. I am kind of highlighting the cell membrane. I would draw an arrow to that and label it cell membrane. Okay, I would also take a look at this dark spot in the middle. Well, not necessarily the middle, but inside the cell, the dark spot. Kind of circular, kind of smushed. That is the nucleus. So I would make another arrow, point at that, and write nucleus. Okay, and then the inside of the cell is made up of the jelly-like substance. You can't really, there's no shape to it really. I would just point to an inner part of this cell and write cytoplasm. Okay? Cool. Then, we're going to look at this one. Oh, sorry. There we go. To zoom in. Now, this is on medium power, so move on. These are cheek cells on medium power. You can see that they don't look much different. They're just enlarged, right? So again, I would draw what you see here. So I would draw these 3 cells kind of quickly. Go boom, like that, boom, like that. These ones are attempting to stay connected. All right, then I would label one of the cells. I would say, all right, here's the cell membrane. This is the nucleus. This is a nucleus. This is a nucleus. Okay, and then I would label the cytoplasm. Okay, so I'll give you time to do that right now before we move on. Oh, I'll get back on that. All right, who needs more time to draw? Okay, Zach, I will give you 1 more minute. All right, I know it's quick, but I want to move on to the onion. Okay, and you can touch up your drawings later, everybody. Okay, so 1 more minute, and then we'll move on to the onion.
All right, I'm done.
Okay, cool. We are not doing high power. Do not worry about high power. All right, just not worrying about it today. All right, cool. So we're moving on to the onion cell. Now I need to get this up. Can you see this? Someone give me a shout out if you can see this.
Yeah.
Okay, cool. All righty. So here we have, believe it or not, this is a piece of onion skin. I want you, see now this is kind of bright, but
Gross. It looks so good.
okay, I gotta let it focus. There we go. All right, so you'll notice, you see all these square-ish, rectangular-ish, more blocky looking things? Those are cells of an onion. Okay, those are cells of an onion. Now this is on low power, so we're not that zoomed in. But I want you to imagine that this is your box that you have to draw in, and draw what you see. Oops, I gotta move it back. Draw what you see. Draw a bunch of these cells. Okay, don't worry about this circle right here, or these circles. Those are just air bubbles from that happens when you have water or a liquid substance in there under the cover slip. Sometimes it doesn't work. So draw what you see. Draw these cells. This is low power right now under my microscope. Somebody give me a shout out, because I can't see anybody right now. I'm looking at a picture of the microscope. Somebody give me a shout out. What is different with these cells already that you can tell?
They're more spaced out, and they're
I don't know. I think they're actually kind of really close together. I see them all running into each other.
yeah.
Okay. What else? Anybody else? What's the difference?
Did you see that she was not able to connect?
The difference I see is that the what's the other cell? The cheek cell, they're in clumps.
Okay.
Like there's pieces where they're scattered,
Sure.
but this one is just whole.
Sure. What about if I say the shape of these cells? How are they different?
They're more squarish.
Yeah, more rigid, more rectangular, more uniform kind of. Sorry, I know that's very bright. I'm working on making it not so bright. Come on. Oh, okay. All right. That is because plant cells. Someone needs to mute themselves. I don't know who it is. Somebody needs to mute I hear some TV. Okay. Plant cells have a cell wall, which is more rigid, made up of cellulose, more rigid, and gives a more of a uniform shape. Okay? So that's why. Hopefully you got that drawn under low power. Then I will go to medium power. Let me get on this again. Here we go. And then I'll have to adjust. There we go. Good. Everyone see that? So we're just
Yeah.
closer up.
Yeah.
You can see all these cells, how they're much more rectangular, rigid. And I didn't even get an awesome piece of onion, but I can tell that I've kind of folded the skin over on it, or it folded over on itself underneath the cover slip when I put the cover slip on. So it's kind of overlapping, but I can see like this is a good one right here. That's a good onion cell. But you should be drawing all of these, kind of making it looks like a honeycomb almost at points. It's difficult to see the nucleuses of these. What you need to be doing is writing about the cell membrane, the cytoplasm. And then I know it's really difficult to see some of these nucleuses. Some of them you can, but point out to a spot and say, hey, here's the nucleus of a cell. Okay, when you draw and label. Okay. We are at about 5 minutes left in class. So hopefully at this point, I know we had to rush through this. We had to go through a lot of logistics, but hopefully at this point, you should have had your problem. I know that. The hypothesis is there for you. Good. Materials are there for you. Good. Methods we are not doing. So methods, I will strike through it. You can strike through it if you want. Okay. Observation. You should have on a separate piece of paper the cheek cell drawn here and the cheek cell drawn here. We are not doing high power. So I would just delete this column. There you go. And then I would do it on this one. I would just delete this column. All right. So then you should have your animal cell drawn twice and labeled, drawn and labeled, and also your plant cell twice drawn and labeled. Okay. Then you go with your summary of results for your conclusion, whether or not this hypothesis was correct, and self-critique. You should understand how to get a 3. Why should you understand?
So
Because yeah, look at this rubric right here. Okay? You can look, okay, is my question clearly defined and related to my hypothesis? Go up and look at it. Check. All right. Is there correct punctuation? Go up and look at it. Check. Then your conclusion. Does it hit all 3 parts and clearly answer my problem? Check. All right, good. Then I would get a 3. Okay. Now you got to be honest with yourself, right? Got to be honest with yourself. Yes, James, the plant cell is the onion cell. Yep.
what if we don't?
Then we would.
What if we don't have a microscope?
I don't know who's said. Yeah. Okay. So then we would move on to science target 2, developing used models. So you had to draw a model. Does your image take up the entire space? That is like the main criteria. Did you draw your image big enough? If you drew it tiny so that I can't see it when you take a picture of it, that's not going to get you a 3. Okay. Then labeling is consistent. Did you label it? Nucleus, cytoplasm, and cell membrane on all of your drawings. That's a big part. Then this is another huge part. there is a noticeable difference between the images in different powers. Sorry. So I should be able to tell the difference between your low power and your medium power. Okay? We good? You all have access to this? Zach, you don't need a microscope, bud. I just did it on the microscope. And you could now do this. Look what I did. I Googled a cheek cell under microscope twice. You could do onion cell. There we go, under a microscope. All right, and does this look like what we saw right here? Kind of, right? Theirs is a little clearer. There. Does this look like what we saw, Zach? Anybody feel there?
Yeah.
Yeah, that looks like what we saw. Cool. So if you have trouble drawing, what we just saw, I know it was quick. You can use the internet as well. All right. Okay. I have a class coming in about 30 seconds. So, well, not 30 seconds, but 2 minutes. We got to wrap this up. What do you got for questions?
None.
Zach says nothing. Okay. I'm going to stop sharing my screen. Anybody else have questions? So that lab report, get that done. Turn it in by, I believe I said the Monday is the due date. Okay. I'll give you till Monday. So if you need the weekend, you can do it. I don't think you should. I think we went through a good majority of it. You need to write a good conclusion. We went through a good majority of this, but you need to write a good conclusion. So that is when it's due, is Monday. Otherwise, I'm not seeing any hands. I'm not hearing any shout outs. I'm not seeing anything in the chat. So I'm assuming we're okay on what needs to be accomplished. Okay, cool. You are free to go. I got to get ready for my next class. Have a great day. Have a great Thursday. Bye-bye.
Mr. Dayton, I listened to this radio station in the morning, and they have this like crazy news segment.
Mm-hmm.
And this guy in China had a heart attack while helping his third grader with math.
Oh my goodness.
What the heck?
When your parents were late, helping with math and then just keeling over. Oh man, that's funny, but not funny at the same time. I'm sorry for that guy. I hope he's okay. But yeah, math makes some people crazy.
Yeah.
James, why is the Hangout still disabled? I don't know, because I think it might have been getting abused last year, where students were chatting with other students and not saying appropriate things. I really don't know. I think we're gonna get it fixed.
That was actually the reason why.
Okay, so yeah, I think that's going to be fixed eventually, where like teachers can talk with you, but I don't know if you'll be able to chat with your friends. So yeah, that's all I got. All right, bye bye everybody. Thank you for tuning in. I appreciate it.
Bye, Mr. Dayton.
Bye bye. See you, Sawyer.